Welcome back for part two of our connective tissue discussion. We left off with our identification of cells, and we're going to pick that back up as we talked about adipose tissue, and then we will move into classification of our connective tissues as they visually appear in various histological slides and different locations within tissues. So with our adipose cells, what we are going to see is certainly they are functioning in the storage of triglycerides and fat, and also in the release of that tissue. And what we see is that fat is largely broken down in the duodenum. This is the first portion of our small intestine. We have pancreatic enzymes that are added into the chyme, this partially digested material in the duodenum, and this is pancreatic lipase, the breakdown of lipids. This breakdown material is absorbed into the intestinal epithelium. It is processed and converted into triglycerides. Now, this happens in the smooth endoplasmic reticulum in our epithelial cells, and now these are going to be transported throughout the body and placed into the serum. But remember, triglycerides are hydrophobic. They're organic, long hydrocarbon chains. So these are going to be packaged into phospholipid spheres that contain a core of triglycerides and cholesterols, and these are called chylomicrons. In physiology, we learned about the lipoproteins, and these are going to be a type of a lipoprotein that we see, this transport vesicle type structure that's used to transport triglycerides. And this is typically what we see, and again, you're going to be familiar with this from physiology as a lipoprotein. Chylomicrons are a type of lipoprotein, just like the low-density lipoproteins and the high-density lipoproteins that we talked about. But in this illustration, we can see our core of triglycerides. We have our outer casing of phospholipids, and this is a single layer, unlike the plasma membrane, which is a double layer. And we have our apolipoproteins. And remember, these are going to be the ligands for receptors to help target these molecules to various locations in the body. Now, we can also recruit triglycerides back out of the adipocytes. And this is going to be a lipoprotein lipase, similar in regards to what we see produced in the pancreas. And this is going to break down those chylomicrons into the fatty acids and into glycerols, and these are going to be able to diffuse into the adipocytes as well. They will again be processed. They are going to be placed in triglycerides again once they're back inside the cell. These chylomicrons and triglycerides are simply too large to effectively diffuse from place to place. That's why you see them packaging. That's why you see them being broken down and used in various ways. And as we see, finally, triglycerides reformed in the adipocytes, which we refer to as fat. Now, we also respond differently to our various neurotransmitters and hormones, epinephrine and norepinephrine. What these are going to do is they're going to activate the formation of cyclic AMP. This is a potent second messenger signaling apparatus. And what this is going to do, it's going to cause the reverse process where we have another lipase, and it says hormone sensitive. In essence, it's going to respond to epinephrine being present. And this is going to break down the triglycerides in the adipocytes, convert them back into fatty acid and glycerol, and this is going to be placed back into the bloodstream that we can use for very rapid sources of energy production. Because when are we going to see epinephrine in the circulation? In a sympathetic response, the fight or flight response, part of our autonomic nervous system. And we need energy, and we need energy immediately. Hence, this hormone-sensitive lipase working to return these essentially starting materials that are going to help us form ATP. So here we see an illustration. We have our hormone-sensitive receptors on our adipocytes. Their cyclic AMP start the signaling cascade. Here we have a triacylglycerol lipase that's going to convert our triglycerides down into fatty acids. And here we see glycerol. This is sequestered in the bloodstream. Again, it's going to be packaged in transport vesicles, going to various locations in the body. Here we actually have a fatty acid transporter We're going to plug this into the citric acid cycle at various ways, and this is an example of in our myocyte. In our citric acid cycle, our oxidative phosphorylation chain, we're going to be able to produce ATP. Looking at other cells that we see in our connective tissues, still staying in that fixed cell category, we have mast cells. Now, mast cells, unlike what we've seen with the adipocytes, they are going to be somewhat motile. They can move around, and we're going to give them different names depending upon where we find them in the body. Now, when we look in connective tissue, these are going to be the largest of the cells that we will identify. And they're going to be characterized because they're going to have a number of really large, dark staining granules, and they are filled with heparin and various mediators. We talked about these in inflammatory responses in physiology. We'll touch on these again as we go through this part of the lecture, the functions of mast cells. So here we see some mast cells, not the best illustration, but you can see all of these large specific vesicles that are present in this cell. I believe this cell has ruptured, but all of these vesicles very closely resemble what you're going to see with these cells. This is in the peripheral blood, actually. And when a mast cell is in the peripheral blood, we don't call it a mast cell anymore. Do you remember from physiology what we call a mast cell in the circulation? We call it a tissue basophil. I'm sorry, a basophil. Out in the connective tissue, it's referred to as a mast cell or a tissue basophil. So from physiology, our understanding of basophils, mast cells, are very clear. These are going to be extremely sensitive to the IgE antibodies because they could potentially have these IgE receptors. And if you bind these antibodies that are produced to various antigens, such as allergens in this case, then that's going to lead to a cross-linking of the IgE molecules and receptors, activation of the mast cells, a massive and rapid release of this heparin and histamine, all of these mediators, prostaglandins, and that's going to lead to an immediate allergic response, which unfortunately inflammation is part of that response. Here we see at the bottom we have two IgE receptors that have previously bound these IgE antibodies in red. Now the allergen in this case has come along, cross-linked these two antibodies together, which pulls these receptors close enough together to signal the activation of this particular mast cell. So here we have our histamines released, we have prostaglandins released, all sorts of interleukins released, growth factors and chemokines released. This is going to lead to that rapid response. And if you have enough of these cells undergoing this rapid degranulation or release of this material, you're going to have a very strong, potent systemic allergic response. And do you remember what we called that response in physiology? Anaphylaxis. And the pinnacle of that is anaphylactic shock. If you don't seek immediate attention and get some epinephrine to try to offset those things, then you're looking at death in a very short period of time. So just a reminder, some of these mediators that we see in the mast cell that are released, we've mentioned the heparin, the histamines, there's some proteases. We have various factors that are going to enhance and increase the effectiveness of the other cells in our immune system, eosinophil chemotactic factor and neutrophil chemotactic factor, to get them on the scene to help eliminate whatever antigens might be present. We see the prostaglandins and leukotrienes. Most of these things, again, are going to function very similarly to what we see with essentially inflammatory type reactions. You're going to dilate the blood vessels. They're going to become more permeable and leaky. All of those things that you associate with inflammation. Now here we have two examples of mast cells. This mast cell is what you typically are going to see in your connective tissue. You can see a lot of very dense granules, not a whole lot on the cell surface, fairly smooth. But in this particular cell, the granules have all moved close to the surface, and many of them have already been released out into the environment. You're going to undergo exocytosis, fuse the vesicle membranes with the plasma membrane, release the components of the vesicle out into the extracellular space. And this term degranulation is a light microscopy term because when you look at a normal mast cell under the light microscope, you see granules. And when you look at an active mast cell, you don't see granules. So the difference is you've lost the granules or degranulated. That's after the secretion of those biologic mediators. So here's a little bit more of the function of many of these mediators. We have histamine. Again, as we mentioned, just think of what happens during inflammatory responses, and most of these are going to hit right there. Vasodilation, increase the permeability. We know the chemotactic factors are going to basically draw these other white blood cells into the area, these other immune cells. Leukotrienes for permeability. Prostaglandins cause bronchospasms. Now, this is a different one in the sense that we're not increasing permeability leading to inflammation, but the bronchospasms are a symptom and probably where you're going to end up in the most trouble physiologically because you can't breathe. This is what we're wanting to offset with the EpiPens and the epinephrine to dilate those airways so you can return to breathing again. PAF, increased permeability. Thromboxane, we're looking at platelet aggregation, which again somewhat seems to be anti-intuitive. It doesn't really go along with our inflammation and the mediators of inflammation. But then we have bradykinin, which is a vascular dilate. Pain sensation, this is going to let you know that in this area something's going on. We don't like pain, but pain's not a bad thing. It lets us know that something needs to be fixed, remedied, pay attention to this particular area. All right, we're still working with fixed cells. Now remember, fixed, it's a relative term. Mast cells can be in tissue, they can be in the circulation, two different nomenclatures and names when we do that. Macrophages are another. You're going to find these doing most of the work out in your tissues, but once these cells are in the circulation, and here you see they're produced in the bone marrow, we refer to them as monocytes. Now, these are the vacuum cleaners of the body. They're going to remove debris, whether it's from damaged cells, dead cells, microorganisms. These cells are going to clean up whatever's present in that environment. But the other thing that we're going to mention about macrophages that we talked about in phys, these are antigen-presenting cells, APCs. So they're going to be able to activate such as helper T cells to really get the immune system rolling. Here we see a nice illustration of a macrophage. This is a scanning electron micrographic view. You can see the macrophage. You can see its leading edge of cytoplasm. It's attached to this substrate, and it's extending all of these cytoplasmic processes and grabbing onto these rod-shaped bacteria that we see, starting to engulf them. The process of phagocytosis, it will remove this material. Now, when you're looking for a macrophage, what you're going to look for, they're relatively large cells, basophilic cytoplasm. Whenever you think of basophilic versus acidophilic, think of pH paper. What color is an acidic pH paper? Red. Basophil, blue. So basophilic cytoplasm means it's going to be somewhat of a dark bluish color. Now, the other thing we're going to see is a nucleus that is not perfectly round. It's oval shaped, not necessarily going to be right in the center of the cell, but the other characteristic of these large cells is they're going to have an indented nucleus or somewhat of a kidney shaped nucleus. Now, within the connective tissue, we're going to see a lot of lymphocytes. We'll talk about them later. But you can see this much larger cell, much more cytoplasm. And the cytoplasm of this is going to have a lot of vesicles within it as well. Some of these may be clear if it has ingested some material in the process of breaking this down. I often refer to these vesicles as a soap bubble sort of appearance to the cytoplasm. Here we see a fairly large oval-shaped nucleus. We see nice round nuclei with these other cell types. And a little bit of an indentation. Usually you're going to see a little bit more of an indentation with these macrophages. In the plane of section, it may not be visible at all, but this is a nice longitudinal section through that oval nucleus of a macrophage. Here's another macrophage. We can see cells. These are plasma cells that we see here, lymphocytes. and then this large cell, and here we've got a nucleus that's just round and in cross section, so we don't see the indentation. But notice how we have this bubbly look to the cytoplasm of this cell. Large cell, soap bubblish appearance, that's indicative of macrophage. You may look at this after we did the multilocular adipose tissue and understand you're not going to have one solitary brown fat cell in a sea of a lot of other cells. If you've got this solitary cell that looks so different, it's likely your macrophage. Now, depending on the location, we call macrophages other things. We already mentioned in the bloodstream, we call them monocytes. Well, in the liver, macrophages are Kupffer cells. In the lung, they're referred to as dust cells, and in the skin, we refer to them as Langerhans cells. Regardless of the location, regardless of the name, the function of these cells are still the same. That's what we've gone over for our generic resident macrophages. This is a light level view of liver tissue, and what has happened in this preparation, they've taken slices of living liver tissue and put on some India ink or some other carbon type material and given the macrophages time to phagocytose this material. So the dark cells that we see in this particular illustration are the cells that were able to engulf and phagocytose the material. These are in fact the Kupffer cells in the liver. If you do the same thing in lung tissue, the dark staining cells that you see in this are the macrophages. So you can see in a lot of tissues they are quite extensive and high in numbers in many places. So macrophages, they can certainly be resident. That's mostly what we've been talking about with our fixed types of cells in connective tissue. But we can also have macrophages that are attracted by chemokines from a great distance. We talked about immune responses. We talked about our short hitters, the first cells on the scene and first to go. You remember those? Those are the neutrophils, and the macrophages are the long hitters. It takes them a little bit longer to get there, and they hang around a bit longer. So those would be the elicited macrophages responding to chemokines and a local site of infection. Now these are weird cells. I'm not sure that you're necessarily going to see these any during the semester, but they go along with macrophages, because if you take several macrophages and you fuse them together, you're going to get this one tremendously huge blob of multiple nuclei running around. And sometimes these are going to be responsive to inflammation. So in areas of inflammation, if you have a biopsy, for instance, from inflammatory tissue, you may see these foreign body giant cells. Now, if we thought macrophages were big, this is probably the size of a macrophage that you would see just a solitary macrophage. Now look at this monstrous cell. I don't know if you can even count the nuclei, maybe 40 or 50 nuclei in this one particular cell. But that's a foreign body giant cell. It has the same function as macrophages. Why they fuse together, I think the jury is still out on that, but we want you to be familiar with those. Now, more transient cells are going to be listed in these categories: the plasma cells, leukocytes, lymphocytes. But again, understand that our mast cells and macrophages are also somewhat transient. They're more resident than they are transient, but they can move around. These are much more motile that we'll see in and out of bloodstream and in and out of our tissues. And we will cover these. The plasma cells, certainly these are going to be derived from our B cells, and they'll produce antibodies. Leukocytes are our white blood cells, and then the lymphocytes, the B and the T lymphocytes. And we'll describe these much, much more once we get to the system, the section on immune system. All right, now we're going to look at the organization and classification of our connective tissue. And this is principally going to revolve around the amount of fibrous tissue that we see versus the number of cells that we see in a particular tissue. And our first classification we're going to look at is referred to as loose connective tissue or areolar connective tissue. And some of the places that you find these is going to be beneath the epithelium of the skin, beneath the epithelium of digestive system and respiratory tracts. We're also going to see the mesothelial lining of body cavities, adventitia of blood vessels, and the lamina propria of the alimentary canal. Now, if some of these terms are not so familiar to you, hang on, because once we get to these different organ sections, terms like adventitia and lamina propria are going to be commonplace for you. So, characteristics. We're going to be looking at ground substance in the tissue fluid, certainly our fibrous and molecular components that we have there. Cells that are present in our loose and irregular tissue are going to be fairly common to what we see with our fibroblasts, adipocytes, macrophages, mast cells. All the usual characters are going to be present in our loose connective tissue. A lot of collagen. Collagens 1, collagens 3, remember, sometimes they're referred to as reticular fibers, and also thinner diametered elastic fibers. Now, as we look at loose connective tissue, it's loose because it's going to be accessible. Transient cells are going to have an easy time getting in and out of that area. We're going to have chemokines that are readily able to diffuse throughout this very loosely woven anchorage tissue. And so when we see this, you can see a lot of cells and you can see the fibers, but there are a ton of cells present in this image. And so the very, very fine fibers that we're going to see running through here are going to be our elastic fibers. And I don't really see a real good one that's a nice large diameter fiber that I would say is a nice collagen fiber, so we're not going to point to those. Much of what you see here is elastic fibers. Now this is really cool because you can see a capillary. Now I know we're not on blood vessels yet, but this is capillary. You know it's a capillary because this blood vessel is only about 1 red blood cell in diameter, and it only consists of 1 cell layer, these very thin, flat epithelial cells. And since we just left epithelium, how would you classify that epithelium? Simple squamous. And when we get to the blood vessels again, remember, we're going to identify this as the endothelium of the blood vessel. Now, you can see this nice stellate nucleus. Once we move away from the blood vessel and we see this oval-shaped stellate type nucleus, we're really having a hard time seeing cell boundaries here. These are going to be the nuclei of our fibroblast as they're running throughout this tissue. The larger cells are likely going to be mast cells and macrophages. Once we find our lymphocytes, they're going to have nice round-shaped nuclei, fairly dark nuclei like we see here. Some of these red cells that you see, that red staining cytoplasm, that is not unlike what we're going to see with our eosinophils as part of our allergic response. Now the second type of tissue is referred to as dense. We have loose, we have dense. So in this case, many more fibers and fewer cells than we saw with our loose connective tissue. Now we're going to subdivide dense connective tissue into 2 categories. One, the fibers are totally random. There's really no organization to these whatsoever. And we're going to see irregular connective tissue, and we refer to this as dense irregular connective tissue. Beneath the dermis of the skin, we're going to find it wrapped around nerves and capsules of organs such as spleen, kidney, and various lymph nodes. And when we look at this, you can see tons and tons and tons of collagenous connective tissue, and you're seeing very few nuclei. It was difficult to count the nuclei in our loose connective tissue, but in this, you can actually sit and count the nuclei that are present. You can see there's much less in the way of space in this tissue, many fewer cells, greater density of fibers, so that is our dense, irregular connective tissue. Dense regular connective tissue, you're going to have even fewer cells than you saw with the dense irregular. And specifically, we're going to see this dense irregular as ligaments and tendons. This would be our dense regular collagenous connective tissue. And when you look at these, you see no space whatsoever. This is a longitudinal section through tendon, and you can see these elongated nuclei. These are our fibroblasts, and really no space whatsoever. In tendon and in ligament, you're going to see very little, if anything, in the way of blood vessels, really nothing in the way of nerves. This is a cross section or a transverse section, Again, you can see how few cells we have and very little, if any, spacing that you see here. Some of the white space that you see in this particular field, these are going to be artifacts of the preparation of the tissue. So this is not real, in fact, space that you're looking at. So no space, tons of fibers, few cells. That is our dense connective tissue.